I'm going to tell you today about something we call nanosatellites. Has anyone ever heard of a nanosatellite? Raise your hand. Okay. Yeah, well, that's because you guys were at Hilton. That's kind of cheating, and I know Alvin has. So nanosatellites, well, a number of years ago, people that built satellites decided that they were going to make satellites that were smaller than typical ones. And the typical ones were the size of a small car. And so they decided any satellite under 100 kilograms would henceforth be called a microsatellite. So it's a different sense in which than which we use micro in microsystems. And then, of course, somebody at university came along with the idea that they would make still smaller satellites. So they decided anything under 10 kilograms would be called a nanosatellite, since the ones under 100 are micro. And yes, there are ecosatellites too. Those are under a kilogram. And they're typically in cube form. You'll see a couple of images of some. These are, some people, although these are not cubes, some people call these 3-cube nanosatellites. They are, in fact, approximately the size of 3 conjoined 10-centimeter cubes. And in fact, our science experiment in most of these occupies about 2 cubes worth, so 20 by 10 by 10 centimeters. And the spacecraft brains, called the bus in the space industry, that has the radios, the batteries, and so on, that's about 1 10-centimeter cube. People do things with just a single 10-centimeter cube, but what they don't tend to do is live biology experiments. We found that the complexity that you need to do something interesting, like looking at gene expression in outer space, requires more than a single 10-centimeter cube in total. So these are big nanosatellites. They typically weigh about 5 kilograms. And we launched the first one of these that we did called GeneSat. You can guess that measured gene expression in 2006. PharmaSat, which looked at pharmaceutical drug and dose response, that was launched in 2009. And the one I'll tell you the most about today is called OREOS. That's the organic and organism response to orbital stresses. That launched in 2010. The science got finished about a year and a half later. It's still up there in space. It'll be up there for another 18 years or so. It's in a rather high orbit. But this one disintegrated after 3.5 years. This one after about 2.5 years. When they come back to Earth orbit and disintegrate, they completely burn up, with the exception of some little teeny titanium screws that hold on the solar panels. And they're really small. They're like 440 screws, maybe they're even 256 if you're a me, you know what that means. Those are little screws. But if you ever see little tiny titanium screws lying on the sidewalk, they could be some of ours. But nothing bigger than that will fall on you from the sky that we put up there now. There's a lot of bigger stuff that falls out of the sky regularly, so don't worry about these things. The reason I say all that is just to emphasize we don't get samples back from space. Everyone says, oh, do you do the analysis in your lab? No, all the analysis is completely autonomous, all done in space, and the data come back transmitted by a little bit of it on this little dipole antenna, which is tuned to a typical ham radio frequency, amateur radio frequency. In fact, we publish on a website what that frequency is, and the ham radio operators around the world can tune into the little beacon signal that comes out every 5 seconds and get pieces of our data. And they can track the currents through the solar panels, the temperature of the satellite, and a whole bunch of other things that we basically publish in real time, if you will. The science data and the two-way communication to the satellite comes down through this antenna. This is called a patch antenna. It's just a square film of gold sitting on top of a ceramic substrate. And this is a 2.4 gigahertz, basically a high-powered version of Wi-Fi. It's the same frequency and the same mode hopping that you guys all use when you connect your laptop to whatever Wi-Fi is available. This one puts out a little more power, so it can talk to us from 650 kilometers away. But the communications link is rather like if you were parked in your car in the parking lot outside Starbucks with truck in between, and you try to get the Starbucks signal. It's not very good. So our data rates are not too high. The links from these radios are not great, but they are reliable in the sense that we're able a couple of times a day to get some data down. So that's just sort of a general introduction. You can see, I had a little help from my friends here. This is a team that worked on the OREOS mission. Many of them have worked on one or both of these other missions. And one thing I'll emphasize is that out of however many names there are here, 30 or something, I don't think there's more than one, at most two, that were full-time on any of these projects. So most of these people are working 2, 3, 4 projects in parallel. And that's what keeps the cost of these low. And when I say low, I mean by NASA standards low. It means sort of ballpark of a couple of $1 million. That's starting from basically a clean sheet of paper, designing the experiment, building the whole analytical system, building the whole satellite, getting it in space, getting all the data back, a couple $1 million. So it's inexpensive for space science, even though that would be a good-sized grant for a lot of basic research. This one was probably the quickest one we've ever done from the start, from our first meeting when we defined the 2 science experiments that would happen in here, until we had our first orbital data back. I think it was 27 months. So we basically created 2 separate instrument payloads from scratch and assembled everything and got it launched in just over 2 years. Okay, so before I go more into the engineering side, I want to sort of give you a little bit of motivation behind why anyone would want to study biology in outer space. And there are 2 sort of branches, if you will, which sound very similar, astrobiology and space biology, but they have a lot of important differences. So astrobiology is very fundamental. It's about the origin, the evolution, distribution, and future of life in the universe. So it's very much asking, where did we come from? Where else might life be able to exist? How did things happen in a very broad sense in the context of the cosmos? So it's sort of broken down into several key areas. One is understanding the details of the distribution of prebiotic chemistry, the chemistry that is the starting point for life. And one of the 2 payloads I'll tell you about today, that's the space evolution of, forgotten what this acronym is for. This is looking at organics and how they change over time in space. So that's one of the experiments I'll talk about. And the second one is studying how life can adapt and survive in extraterrestrial environments. And the second payload will look at that one. So you'll hear about these 2 rather different experiments. One is really more astrochemistry, and the one really does deal with how biology survives in the long term. Also, looking for signs of existing or extinct life is something that's part of astrobiology. And our SIBO payload does that to a limited degree. It doesn't actually search for organisms. What it does is try to inform us about how long organic molecules that might be biosignatures can survive. So things like porphyrins and quinones. How long could they last if they were left on the surface of Mars or on an asteroid by life that had once been there? How many years or decades or centuries or millennia could you expect them to survive? There's none of this that we do in the current experiment I'll tell you about today, but another part of astrobiology is looking for habitable environments. Obviously, you've heard a lot of the news about this recently, about planets that look like they have environments similar to Earth, and the Kepler mission is finding those more and more. So this is all about fundamental understanding of life in the universe. In contrast to astrobiology, fundamental space biology is much more applied in a sense. It's the effects of the space environment on terrestrial life, and very much with an eye towards humans being explorers of space and how space will affect us and organisms that we take with us. So the 2 main effects are first microgravity or reduced gravity, depending on the situation. And in mammals, that changes fluid distribution, loading of your skeleton. It creates immune stress, which is significant. Bone density changes rapidly. Your pelvis and the load-bearing bones in your legs lose bone density at a rate of about 1.8% per month. So imagine osteoporosis, 2% a month in the initial months that you're in space. So all of us, as we age, eventually start to undergo osteoporosis that can become an issue. This, if you will, is an accelerated testing platform for osteoporosis. And some things have been learned about that in the space studies that may be relevant to many of us. Cells and microorganisms also are affected by reduced gravity, but it has more to do with transport of nutrients and waste than it does with the skeletal things, though there are actually skeletal factors. The other significant parameter that's different in space than on the ground, of course, is radiation. And damage from high-energy ionizing radiation particularly starts to happen outside the magnetic shield of the Earth. And so that means you're more than about 70,000 kilometers away from Earth. It's obviously a very gradual difference. DNA damage is one of the primary effects, and we have an upcoming mission to look at that. DNA can be broken, cells can die, mutations are possible, and that's one of the things that's a concern for long-term habitation in space. There are other things in extraterrestrial environments if you're not just in a spacecraft, but if you land on the moon or Mars or somewhere else, even an asteroid is being considered. The dust, the environment can chemically do interesting things. And so all of these things can potentially interact with one another, and sometimes they're antagonistic, sometimes they're synergistic. So the why of all of fundamental space biology is certainly human space travel, thinking about perhaps inhabiting the moon with base someday. Planets are obviously much further off if they're even practical. But also back on Earth, insights and therapies for human disease, for aging. Radiation effects sounds a little esoteric, but many of us, sometime point in our lives, may develop cancer, and radiation is a very common way, still quite effective way to treat many sorts of cancer. And so any of us who has that kind of treatment might be concerned about the biological effects of radiation. So this is simply to say, as we all say in an academic environment, you study basic phenomena and you learn new things, and that informs you and helps you answer lots of questions and problems. So here's some examples of a couple of these CubeSats. These are the 10-centimeter cubes that was from Cal Polytech in our neck of the woods. This is from the National University in Taiwan. This is GeneSat, the first one we flew, 4.4 kilograms, 3 liters roughly. So why do we do things in these small sats? Well, partly because we can. All the sort of miniature micro nanotechnologies that probably all of you are engaged in one way or another, you kind of see a laundry list here, allow us to do interesting science in something that small. And I don't think that's a surprise to any of you because you're doing lots of things on a small scale. There's a lot of systems integration here, somewhat more than you might have to do for a lab experiment, but the basic building blocks are very similar. The most important reason probably is access to outer space. You can get these into space for a few tens of thousands to a couple $100,000. In fact, if you work at a U.S. university or a U.S. national laboratory, NASA now has a program that will put them in space for you for free. So if you have a proposal to do something interesting from a science or engineering perspective in outer space, and you have the means to develop your own satellite, and as I said, $2 million is a NASA budget, you can do less ambitious, simpler things for a lot less than $2 million, then NASA will put this in outer space for you.
So how does the launching process for this is working for you? Where is it actually taking place, and what's the procedure?
So various places. NASA, for this program that I was talking about, buys space on various space launches. Our first three went up on U.S. Air Force launches. There we actually had to transfer money to the Air Force, a couple $100,000. But our more recent one went up on an ISS resupply mission operated by SpaceX. So NASA basically bought some launch slots or deployment slots on that SpaceX mission from SpaceX. So it's some combination of military and U.S. government launches, and a lot have gone on also European launches and Russian launches. NASA actually shops around. I think they only use U.S. vendors so far for their program, but people around the world. So probably more than 100 universities building these things, and probably half of them by now put something into outer space. So there's lots of ways up there. So they're a great education vehicle and sort of within a budgetary range where that's feasible. You learn a lot about systems integration as we discussed today, but there's lots of other engineering technologies that go into this, obviously, and you can study some very interesting scientific phenomena. Autonomy is key to these things, especially these that have no tending and have to telemeter all their data back by radio. And a lot of the technologies here, we're starting to migrate to instruments that go to the International Space Station. I just finished a proposal with colleagues to take one of our instruments from OREOS and send it to Mars to look at reactivity of the Martian atmosphere. So these small instruments can find their way into other space missions. So let me give you a quick tour of some of the different kinds, sort of sorted by organism, of missions that we have flown or we will fly in coming years. So E. coli was aboard GeneSat. I already mentioned that. In a couple of years, we will launch ECAMSat. That's the E. coli antimicrobial satellite. That's looking at antibiotic resistance of a uropathogenic strain of E. coli challenged by gentamicin. So we look at various different gentamicin levels. And we have both a wild type strain of the uropathogenic E. coli and a mutant strain. And the mutant strain has the RPOS gene turned off. And the RPOS gene, according to A.C. Mateen, who's the science PI at Stanford, is associated with resistance, with antibiotic resistance. And in fact, antibiotic resistance seems to be triggered by microgravity or by model microgravity that people have studied. So the hypothesis is that this triggered antibiotic resistance will happen in the wild type in microgravity, but won't happen in the mutant in microgravity because it doesn't have that gene. And that's what we'll look at in this mission. Saccharomyces cerevisiae. Anybody know why this is the most important organism on here? Anyone speak Spanish? What's the Spanish word for beer? Yeah, okay. So cerevisiae, and Saccharomyces has to do with sugar. So this makes sugar into beer. This is brewer's yeast. That's why it's the most important organism on the page. But it's a fungus. It's a very well studied model organism. So we send the Cerevisiae organism into orbit in 2009, and we looked at its response to an antifungal drug at various different concentrations, and I'll show you some results from that. In a few years now, we will look in a spacecraft called Biosentinel at a very different sort of biological process, but again in the same organism. That's DNA double-strand breaking repair. So we're looking at that in the context of radiation damage to DNA that occurs in deep space. Bacillus subtilis is a spore. It's a bacterial spore, and that flew into OREOS that I'll tell you about today, and there we looked at the survival of them. We put in a proposal recently, which may or may not get funded, to use this same organism and study mutations in the context of lithopanspermia, which basically means that organisms could get trapped in a crack inside a rock and transported from one body to another, whether it's in our own solar system or another planetary system that might exist out there. People have long speculated that life might have moved from Earth to Mars or Mars to Earth this way by asteroids striking the surface, picking up a piece of rock that had something live trapped in cracks in the rock, and off it goes into space and lands on another planet. There are issues with the heat of entry into the other planet, but I think Earth, Mars, Mars, Earth, it's feasible one way and not the other is the current belief among the people that model this in terms of the thermal environment. But then you start talking about Europa and Enceladus, and that probably is quite feasible because they have no atmosphere to speak of if it gets there quickly enough. So it's just a very interesting fundamental question. Can you move life around in a star system in this way? Ceratopterus ricardii, these are plants. They're the spores of aquatic ferns, actually, very ancient plant. And in Sporesat 1 and 2, we're looking at gravity sensing. We demonstrated last year that we could operate a microcentrifuge that's about that big in diameter. Actually, we had 3 of them in there. And look at calcium ion selective electrodes to monitor the current that comes out of the ion current that comes out of the ion channels in these spores. Basically, they have a mechanoreceptor in them that detects gravity. That's how they decide to send their stems upward and their roots downward. And when that's triggered, calcium ions start to come out of the direction that the spore perceives to be up, and calcium ions go into the direction it perceives to be as down. So these are about 150 microns across, and we have a differential pair of ion selective electrodes at the top and bottom. Top and bottom in this context being towards the outer circumference of the rotating disk and towards the center of rotation of the disk. So when the disk spins, that's how we make up and down in space in microgravity. and that's what we hope to measure in a couple of years. And we'll have both a mutant and a wild type of that too, one that's got enhanced calcium ion currents and one that has the normal wild type. And C. elegans, very well studied model organism. We built a dual wavelength fluorescence imager, which is actually waiting for a flight. That one's not scheduled yet. But all the black ones have already flown in space. The blue ones all have scheduled launches. This one is just a proposal at the stage. This one is finished, but needs a launch. So a lot of interesting science gets into space this way. So I have a couple of slides to tell you about the GeneSat and PharmaSat experiments, kind of a one-slide overview. So GeneSat was our first experiment of this type. It grew E. coli in a dozen microwells on a microfluidic card, basically a laminated acrylic multi-layer fluidic card. The little white circles you see are nylon mesh filters that prevent E. coli from getting washed out of the wells because these are non-adherent cultures. So they have to be delivered about 6 weeks before launch. So they know we put them in a state of stasis by just putting them in a buffered saline solution. And once they get up in space, about 6 weeks later, then we replace that saline with a nutrient medium that contains sugar. E. coli take off and grow. And we look at green fluorescent protein to look at expression of a constitutively expressed gene, and we look at optical density to look at cell population. So that was launched in December of 2006. The data came back a matter of days later, and these are the fluorescence versus time curves. Time goes, it's hard to see, but it goes out to about 50 hours here. This is what it looked like in space. This is what an identical experiment looked like on the ground. So you see they're very similar, but there are some differences. The blue curves versus red are two different strains of E. coli, so obviously the blue ones grow faster, and that's true, same on the ground and in space. If we look at the region of the growth curves for these where they're growing exponentially, and put this on a semi-log plot and get a doubling time, they're doubling about half as fast in outer space as they double on the ground. So they grow slower. And that's presumably because on the ground, anytime you have even a very small thermal gradient, that causes convection. So the warm fluid rises, the cold fluid sinks, and there's a little bit of stirring. We don't intentionally stir these, but there's a little bit of fluid movement.
And there's no flow for the nutrition. The solution is...
There's an initial flow to start the process, but the flow is done right here at the time. So there's no continuous
There's
flow.
no continuous.
And the plateau, in fact, the stationary phase occurs when the organisms have exhausted all the sugar in the nutrient medium, and then they stop doubling. And actually, we watched this for a while. The fluorescence stayed up for quite some time and eventually went off. And then in one of them, we had, like, 3 weeks later, a big spike in fluorescence. And one of the biologists theorized that it might have been the cells spontaneously lysing, because some of the fluorescent proteins are partially quenched inside the cells. You let them out, and their quantum efficiency actually goes up. We're not sure, but the main thing was to see that they're growing and metabolizing about twice as fast on the ground as in space. So not a big surprise, but this was really a demonstration that you could measure these kinds of things. Okay, PharmaSat was a drug effect satellite, as the name suggests, and looking at the susceptibility of Saccharomyces cerevisiae to an antifungal drug, voriconazole, if you know antifungal drugs. And this is, again, an optical measurement using LEDs. I'll show you the optical setup a little bit later. And a viability dye that changes color from blue to pink. So this helps us track the metabolism a little more accurately. We have more wells in this one now. There are 48 fluidic wells in 4 rows. And each row gives us a different concentration of the antifungal, and the 4th row is a control with no antifungal. So this way we get a dozen wells of each condition, and that gives us a little better statistical plan. For the electrical engineers in the group, if there are some, this is one of the 2 optic circuit boards that lines up with the fluidic card. And if you look at these up close, they sort of look like the density of the circuit boards inside your cell phone. The electrical engineering is very impressive that our team puts together for this. It's really a high density situation. Okay, so this gets integrated with various fluidics, and again, the size of this whole thing is about 2 liters. Then that gets integrated with the brains of the spacecraft, solar panels on the outside, it's ready to go in space. This one, actually, yeah, you can see the rocket. This is a Minotaur 1. It's a leftover intercontinental ballistic missile. Under the arms treaties that we've signed with Russia, we have to gradually destroy a lot of our stock of intercontinental ballistic missiles, but we have years and years apparently to do that, and so do they. So these are used very successfully by the Air Force to put all sorts of military payloads, non-nuclear of course, into outer space, and they count as having been destroyed. And some of them carry science payloads along. So we were mounted on, it's up in here somewhere, on the side of the second stage rocket motor under the fairing, and which made me a little concerned because I was worried about barbecued E. coli. Rocket motors sound like they get hot. They said, nah, don't worry. The second stage motor only has to burn for about 30 seconds and it won't get too hot. So that wasn't completely reassuring. So we got some thermal data from the people that fly these things and know them well. They actually did have temperature measurements on the outside of the rocket motor, and we plugged it into our thermal model. And the prediction was that inside the payload where the E. coli, or where the Cerevisiae are, the temperature would go up by about 5 degrees Celsius by the end of that second stage burn, and that was perfectly okay. So it starts out cold. So apparently that was true because look, we got data back from outer space. So this is the experiment run on the ground, this is run in space. The color coding is for the different concentrations of the antifungal drug. So blue is a control, green is the low concentration, and obviously the low looks a lot like the control. Yellow is a medium concentration, right around what people would call the minimum inhibitory concentration of this antifungal drug, and red is the high concentration. And so you can kind of see they cluster in the multiple curves. There's a curve for each of 6 wells here.
So what's the y-axis?
So the y-axis is it's an optical signal, and it's the optical signal of a dye called Alamar blue, which changes from blue to colorless. So this absorbance up here, which starts somewhere around, I think, 1.5, is when there's a strong blue color, and then the blue goes away, and this is colorless down here. So we're just looking at the disappearance of blue color. We use that dye because we can count organisms also, but it's not as sensitive. You don't get the signal to background that you get by looking at this viability blue color. The interesting thing we saw here was a lot like what we saw in GeneSat, actually, and that is, again, if you look at the exponential phase of growth somewhere in here, and in there, and there, the ones in space grow about half as quickly as the ones on the ground. Except for, you might have noticed, the red curves here with high concentration of the antifungal. On the ground, the growth is pretty much totally inhibited. There's a tiny bit of metabolism, but not much going on. In space, they are metabolizing. And we know from our optical density measurements and some other experiments that while these kinds of exponential curves that you see in the yellow and the green and blue mean we're getting cell doubling, so that the yeast are budding and the daughters are coming off here, the red curves are indicative of metabolic activity but not actual division of the cells. So the antifungal has inhibited it enough that they don't bud, but they still metabolize. And again, so there's this interesting difference where on Earth the metabolism is almost totally suppressed. In space, there's more of it. And again, we rationalize that this is almost certainly due to mass transport differences, that the voriconazole is not delivered as effectively as it's depleted to these guys, and it's delivered more effectively to these. And so these are better inhibited. We'd have to do some more experiments to prove that's true, but it makes sense in what we see. Okay, so the next thing I'm going to talk about is the OREOS mission. And oh good, there are the acronyms defined for you. So we looked at how the space exposure affected over as long as a year and a half organisms and organic molecules. We looked at their survival and growth and metabolism of this spore, Bacillus subtilis, using CESLO. And for the organic molecules, we looked at UV visible near IR spectroscopy to look at changes in the organic molecules and biomarkers. And the changes were caused by solar UV. More about that in a second. So this launched from Kodiak, Alaska. you know, getting the path ready for transducers, I suppose, checking out Alaska in 2010. On a Minotaur 4, that's a bigger version of intercontinental ballistic missile. The 4 means it has 4 stages, although I think this one had 5, but it must have been a 4 plus. And since it was in Kodiak, Alaska, there are a lot of fences around to keep the bears away. The NASA Ames center director went up to watch the launch, and they sent someone with him to watch the center director, so he didn't get outside the fence and run afoul of one of the bears. This is the flight prototype. This has 2 completely separate 1-cube experiments, and then there's a spacecraft bus, again, very similar to the other 2. This is in orbit today at between 620 and 640 kilometers, and will be up for, well, it's been up 4 years so far, so for another 18 years or so, it will be up there in space. Maybe it's good to point at this while you've got a nice photo here. This carousel has 24 organic samples, so this is the SIBO experiment, and these samples are exposed directly to solar UV all the way down to the Lyman alpha line at 124 nanometers, so they're getting some very energetic photons, and they sit behind magnesium fluoride windows, which actually transmit in deep UV all the way down to that wavelength. The other payload, which is sort of hidden behind solar panels here, you can see it there before the solar panels go on, the one called CESLO, Space Environment Survival of Living Organisms. That's the one where we're doing a bioexperiment. So I'll tell you more about those experiments. So this is kind of the architecture, a cartoon to help you understand what's in each of these. So there's the bus. We'll get to what's in there in a moment. The bio payload, CESLO as it's called, 3 independent biology blocks, each of those with a dozen wells, to grow organisms at 3 different time points, time 0, 3 months, and 6 months, and measure their optical properties, very much the way Pharmasat did. So we looked at a couple of different biological specimens. Only one of them behaved well and grew nicely for us. That was Bacillus subtilis. The other one not so well. Looked at 3 growth initiation times. The other payload, SIBO, looks like this. Schematically anyway, 24 samples up here I mentioned are out in the sunlight. There's a stepper motor that, on command from the software down here, rotates this around. There are a couple of fiber optic pickups that collect optical signal and send it down here to a derivative of an ocean optics UV-vis spectrometer. So we get a full spectrum of these films once a week, and we watch them degrade over time as they're getting fried by the sunlight and solar panels on the outside. So this is a little more instructive solid model than that. Again, the bus is down here. There are some batteries. There are some more batteries. These are standard lithium-ion batteries. This is a so-called S-band radio. This, again, is souped-up Wi-Fi. This is a 2.4 gigahertz, the same thing in the wireless around here. And that sits inside. This is the antenna that transmits that. And then this is the ham radio frequency 933, or is it 433? It's 400 something megahertz, where we publish all the details and people can tune in on this. And Santa Clara actually does all of the, Santa Clara University group does all of the data linking for us, gets the data down. Here are the 2 payloads I just mentioned to you, more on those later. You see there's some radiation sensors on there. We had to add this tail, the so-called nano kite. This is a little structure that looks sort of like a Japanese lamp that hangs in the corner, or a box kite, which is why I named it the nano kite. There's an internal spring in there. These are mylar sheets with a coating of germanium. And this thing all compresses down like a jack-in-the-box till it's only about a ½ centimeter thick. The purpose of this tail is to increase the aerodynamic drag, or change the ballistic coefficient of the satellite. And remarkably, even up at 650 kilometers, there's a little bit of atmosphere. And that is the main mechanism that causes the orbit to decay for this and other satellites. And there is an international agreement between NASA, Russia, Europe, Japan, Canada, and really the whole UN eventually signed on, that all ordinary space missions, and especially these pesky little ones, will come down out of space within 25 years. Our initial calculation said this would be up for 66 years, and we knew we'd have trouble getting an exemption for it because we're not all that important on the scale of what goes into space. This tail, by increasing the drag, the calculation said it would come down to 22 years, and that was good enough. So that's a neat thing that one of our grad students whipped up in a couple of weeks when we realized we needed to address that. These are the two antennas named mustard and ketchup. Don't ask me why. At Santa Clara University. They're about 3 meters in diameter, so they're not all that big. And these talk to the S-band antenna from space, both controlling the spacecraft and getting the data down. There's a little bit of pointing that's necessary so that this antenna is generally facing towards these antennas. If it were pointing exactly in the opposite direction, you'd never be able to close the radio link. And that's done with magnetic rods that are inside the spacecraft. So there are a series of magnetic rods that point along the long axis of the spacecraft, and they cause it, when it's over the northern hemisphere, to point the end with the radio antenna towards the north magnetic pole. And that keeps the patch antenna pointed generally in the vicinity of this antenna, which is at about 38 degrees latitude in the San Francisco Bay Area, so that we can talk to it. So that's kind of a cool attitude control that takes no power and keeps the satellite pointed the right way, as long as you don't put the magnets in backwards. So in space where this satellite was deployed, up at 650 kilometers, 72 degrees inclination basically means it's orbiting between the Arctic and Antarctic circles, roughly, if you superimpose the orbit on the Earth. It doesn't quite go over the poles, but it kind of goes like this. And as it's doing that, it actually gets to regions where the magnetic field's not very strong. You know, the magnetic field of a dipole sort of looks like these curved structures. These curved things, however, are actually cartoon representations that are roughly to scale, well, to each other, not to the size of the Earth, of the Van Allen belts. So the red is the inner Van Allen belt, and the gray is the outer one. The orbit we're in actually takes us through a fairly thick part of the inner Van Allen belt on essentially every orbit. The orbits are about 97 minutes. So every 45 minutes or so, it's going through quite a bit of radiation. And as a result, the radiation environment on the OREOS spacecraft is about 15 times the dose rate that's received at the International Space Station. So it's a pretty high radiation environment, yet we use commercial electronics. These are not radiation hardened. We did test virtually all the electronics to see that they would survive the kind of doses that this would get in 6 months. It's not predicted to last for 4 years, but there you go. It's still, most of it's still working, and it still does regular communications with us. So the biology gets exposed to about 1.3 gray. If you know radiation doses, that means anything to you. That might be typical if you've got local radiation in a limb or something. You might get a kind of localized dose. You wouldn't want it over your whole body. And most of that is trapped protons that are part of this inner Van Allen belt. The organics, which are hanging out there in space to get the direct solar UV radiation over as long as 18 months, get something like 3,600 hours of direct sun exposure. And they also get a lot of ionizing dose because they're not as well shielded as the others. So they get 55 gray, which is quite a significant dose. We have some onboard rad FETs to measure the radiation dose. A number of other sensors too that are very standard. Some MEMS, off-the-shelf devices, some non-MEMS. A MEMS pressure sensor from Motorola, temperature sensors for analog devices, relative humidity sensors that come from Sensorion, radiation sensors, I've mentioned those from REM Oxford in the UK, and various optical sensors for light. These actually are great. We use these in our analytical system. It's a little ASIC that converts light intensity directly into frequency, so you don't need a separate A to B converter. The frequency output you can feed straight into a microcontroller, and it just counts the pulses. And they're linear over like 6 orders of magnitude, so very nice sensors. Okay, so a little bit on the two payloads to wrap up here. CESFLO, again, is the biology payload. We had Bacillus subtilis, and we had a halophile that likes to live in salt flats. This one grows in 3 days. That was fine. This one wants to grow in 3 weeks. That was not so fine. We could really never get them to grow very successfully in the lab or in space. But they forced us to fly a few wells of it just to see if we might get it to grow. We measure red, blue, and green transmittance. This is just what Pharmasat did, and I'll show you some optical curves. Excuse me in a minute. So the sensors I already talked about. This is the cross-section of one of the fluidic wells. The Bacillus subtilis going in dry form. You have to keep the wells fairly small in diameter so that when you add the fluid, it will displace all the air without leaving bubbles trapped behind. It does that pretty well. So the air comes out through something that's essentially Gore-Tex on the top here. And we have 48 of these, sorry, we have 12 of these wells per biounit, so 12 run at each time point. The next sort of level of engineering is the thermal system, and that's a plate of aluminum with Kapton tape on it. You might know this as flex circuit. It's inside all sorts of things, including mobile phones. And this has a pattern of nichrome heaters on it, and the heaters maintain the temperature of all the wells very, very tightly with some integral temperature sensors to a few tenths of a degree while it's in space. Then the optical system looks a lot like what I've seen in your lab, on the poster at least. 3-color LED at one end. Light comes down through the micro well to the organisms. 3 colors are turned on in turn, so we measure the red and the green and the blue. Here's that intensity to frequency detector that I mentioned. So there's one of these, dedicated one of these under every well, so that there are no moving parts. We don't have to move the devices around to measure the various wells. So here's a top view of these 3 blocks, so you can see the layout of the wells here. 6 for one organism and its growth medium, 6 more for the other organism and whatever its growth medium may be. Pumps for both of them, I'll mention those in a second. And there's 3 different time points. So we did these at time 0, 3 months, and 6 months. And the wells are big by microfluidic standards, 75 microliters. With biologists, those are very small. They always fight with us. We say, no, it needs to be smaller. They say it should be bigger. They want 200 microliters. We say 2 microliters, and somehow we compromise here. So here's a solid rendering of what the block looks like. You can see it better that way. The pump system I really like. It's a cool one that Matthew Puccini came up with. This little tubing leads off to an air pump. It's the world's smallest aquarium pump. It's about that big. And the aquarium pump provides pressure down here. There's a distendable, very flexible, I think it's a chloroelastomer, maybe it's a silicone membrane. When the reservoir is filled, the membrane is pushed all the way down in here, and all of this contains the growth medium. The air pressure pushes on the backside of the membrane, and that pushes on the fluid, so there's fluid pressure. This is an electrically operated solenoid valve. So whenever this valve clicks open, and that happens maybe 6 months after we put the whole system together, the pressure of fluid in there, and we also have the little air pump running, will go and fill through the laser cut layer here into all the wells and fill up the wells, and this one fills the other row of wells. So that all happens autonomously in space. All right, here's some more solid models showing putting this together with the radiation sensor on top, and those are the valves sticking out. There's 3 of them now grouped together. You can see this is starting to look about like a cube, and that's now roughly a 10-centimeter cube with everything in there, all the electronics, all the optics, the pump, the valves, all of that fits in one 10-centimeter cube. We thinned this region of the aluminum containment vessel to let more of the radiation through, because this shields quite a bit if you don't. Biocompatibility was a big part of that. You have to test with, for contact with media, all the different materials, direct contact with the cells, the materials they'll come in contact with. And this was a big part of the biology program. There's something you can do to increase your odds a little bit. That is put some activated carbon cloth in. We put that in all our biological payloads. The polymers, the adhesives, and so on, we're very careful about which ones we use, but still they sometimes have some organic molecules that they off-gas, and this soaks them up very effectively and keeps them from doing some impact to your biology. These organisms have to last, as long as, for this mission they lasted up to 1 year from when we first loaded them in the lab till we did our last time point in space. For another mission we're doing in the future, it'll be more than 2 years. And things that you never think of as being bio-incompatible in the lab turn out after they've been in contact for 2 years not to be so friendly anymore. I'll just give you one example. If you do engineering polymers involved, Delrin. Delrin is great, usually with biology. After a few months, cells don't like it. They just start dying. So no Delrin in here. Peak is fine. Just weird things like that. You never figure out if you use things. Our optical system measures at red, green, and blue wavelengths. That's all this shows. And we have this dye in there that's blue to start with. It looks like that. It turns pink over time. And then a little later, after it gets further reduced, it turns colorless. So the red light is very good at being absorbed by the blue color. The pink color is nicely absorbed by the green, the green light's nicely absorbed by the pink color, and the blue light sort of passes almost with the same small amount of absorbance through both. And so these two let us look at how much of two forms of the dye, the unmetabolized and the metabolized, are present, and this lets us count the cells. And so I think this, I'll show you some data curves again from space compared to the ground. Space flight are the darker curves, ground are the lighter curves. This is the 525 nanometers, so the green wavelength, hence the green curves. And what you're seeing here is the interesting shape of this over time, and this is about 12 hours, is the blue turning to pink, that's this coming up, the pink then turning colorless, it goes back down there, so now the blue's gone and the pink is gone, and then the optical density of the cells which are still growing comes up, and that's what's going on here. So it's an interesting shape, and it's very characteristic, and you know this isn't some weird temperature effect, because first of all, it does the same thing in space and the ground. Again, the space growth is delayed a little slower than on the ground, but it basically does the same thing. The mutant looks a little different from the wild type. In this case, the mutant is a radiation-sensitized mutant, so it's more susceptible to space radiation. And I thought, okay, these curves are pretty crazy looking, but interesting. And they're probably different, but you can see the error bars here, the gray sort of things are standard deviations for N of 6 or N of 5. And they do overlap, although in some regions they actually don't. So you really think these probably are statistically significant differences, maybe. Certainly it looks like they are there. So that's time 0. This is after over 3 months. And I thought, okay, somebody screwed up and we've got a copy of the same data again. No, this really is the same data, space flight in the ground for the mutant, space flight in the ground for the wild type, and they really look this much like the time 0 data. So that was impressive that the system worked that way, and that the bacillus subtilis were completely laughing about being in outer space. They didn't think anything of the radiation. But we taught them a lesson. So at 6 months, this spacecraft was in a really interesting environment, and somewhere after 3 months and before 6, he went through a period of severe sunburn. It got the orbit, the amount of sun exposure in the particular orbit this is in varies with time of the year. And it got in a part of the orbit where it was in the sun 24 hours a day for about 5 days in a row. And it got hot. And our thermal engineer thought he had this figured out. He was close, but he was a little bit off. And this is, to be fair, this is really hard to do to get this just right. And so we cooked the biology, not with the rocket motor, but with the sun. So they got up to, our estimate is about 44 degrees C. Now the cool thing is they actually still did grow. We had the thermal data, at least some of it from space, and we tried on the ground, sort of a delayed manner, to also cook the ones on the ground. We obviously didn't bake them as well, because the ones in space, they were just getting going, and this looks like maybe they were totally dead, but if you keep following this, the curve in space actually does eventually do that. It has the same characteristics, it's not as big. They really look like they've been beaten up. The ones on the ground, you can see these are slowed down compared to that, but we didn't roast them nearly as well. It could be that the radiation played a role in this, that it was the heat and the radiation, but it's very hard to say that definitively, so I'm not claiming we know that was from radiation effects, probably mostly. And the mutant pretty much did the same thing. But it's kind of neat to see these kinds of changes. This is just one of the other colors. This is the red light. It shows the blue color going away and the organisms growing up. Eventually we can do some spectral deconvolutions of these and extract a little more data. That's the story for now. So let me tell you in the next few minutes, since Ian said I could go until 7:30, about what the pseudo payload, the space environment viability of organics looks like, which is this will just be a few minutes. So we looked at 4 different molecules. We looked at a polyaromatic hydrocarbon. We looked at an amino acid, and these 2 are kind of representative of what people would call prebiotic chemistry. Now, we don't know for sure that polyaromatic carbons have ever been involved in building life, but they are a great carbon source and they're very plentiful in the cosmos. So they're a potential source of carbon that, if it were further modified once it got to Earth or somewhere else, could be a starting point. Amino acids, on the other hand, we know those are useful in making life, and lots of those come in asteroids in very significant quantities and comets. even sometimes with slight chiral excess, which is another story. These two molecules, though, are a different category. These are biomarkers. So this is iron tetraphenylporphyrin. There's the iron in the middle. And this is amphoruphin, which is a quinone. So this is an electron transfer molecule, and this is an oxygen or carbon dioxide carrier, both very similar to the heme site in our blood, red blood. So the idea is to see how long these things survive in space. So we exposed them for, actually this says 6 months, eventually we ended up exposing them for 17 months and so on, 3,800 hours. And we had them in contact with various interesting surfaces and microenvironments. We had some that had a humid environment, sort of like Mars is at certain times, others in contact with CO2 and so on. We measured initially every day, and then eventually bi-weekly, and then eventually monthly, the UV-Vis near IR spectrum for all of these organic films. We use sun as the light source for spectroscopy, and you can calibrate that with solar features and various sensors in there. Never mind those. So here's a schematic first of a little microcells that carry the organic films. That's the film that sits on a magnesium chloride window. Sunlight comes down through a little diffuser baffle thing, and that goes right through the organic film, down through this metal part of the housing and into a fiber optic pickup. And everything is hermetically sealed. These are indium gaskets that you can get cold weld at room temperature. So the atmosphere in here is quite well hermetically sealed without any heat, so you don't mess up the organic film. And the seals last for literally years. And the fiber optic coupling leads off to this UV-Vis near IR spectrometer, which looks like that. As I said, it's very similar to the ocean optics one, except they are very temperature sensitive, and this has no temperature sensitivity to speak of, from like minus 20 to plus 40 degrees. You almost can't tell if you choose the temperature. So 24 of the samples are arranged in this rotating carousel, a couple of sun angle, sun intensity sensors, radiation sensor, and it's set up in such a way that at any given time, there are two of these fiber optic pickups, but one of them will be staring at the back of the sample wheel, and the other one will actually be looking at a sample. So this was our way to avoid having to use an optical switch. Sorry about that. Again, nobody wants optical switches for anything. But we didn't have room for one. We just used the stepper motor to align this so that you're either looking through a film with this one or you're looking through a film with that one, and never both. And there's actually a position that this cartoon doesn't show well, where they can both look at the back of the wheel, and then you get a dark
What kind of a tolerances do you
the
encounter here with this stepper motor?
Yeah, let's see, the stepper motor, I think it's, how many is it, 720 steps, or was it 1440 steps? So it's a couple of steps per degree,
Okay.
and it's pretty much good to one step, and not much, magnetically it tends to hold in the step that you step into. I think it's 2 steps per degree. We did look at some that were 5 steps per degree. If you want to know, I can tell you which motor it was. It's quite good. Just for scale, the diameter of this is, well, you see it here, 7.5 millimeters there. So you need to position it within about 0.5 millimeter to 1 millimeter. And that was not a problem. Spectrometer weighs about 300 grams. Some solid models, and again, a sample cell. You see 24. Now you can see where you align the two pickups. And if you rotate the wheel around so that they're looking there and there, then you get just dark coming through rather than sunlight. Sorry, the pickups are on the other side. These are the diffusers on the top side. They're the pickups on the other side. There's the radiation sensor. That's a photograph of it being tested on the ground. Our spectrometer builder actually is a guy named David Landis, who was at Ocean Optics for a long time, and then went off and started his own company. Now he's with Draper. And he did some things to flatten the response. This is the solar spectrum. It gives you lots of light in the yellow-green, not so much light here in the UV, where you like to really look at the spectroscopy, and not so much out in the infrared. The spectrometer throughput is actually good. It's got more throughput down in the UV. That's the way we designed it, what he did. Selective rating. And then the detector sensitivity. These are detectors that are used in fax machines, so they don't really care about UV, but they put a coating on them to make them more UV sensitive, so they get a bit more sensitivity here. So David added a neutral density filter, which knocks down the intensity here a bit and lets through all the intensity there and all the intensity there, just to make things a little flatter optically, so that you're not saturating the detector at these wavelengths and bottoming out in those that it doesn't make it perfectly flat, but it makes it more flat. So here's some data from space. Actually, pre-flight, the bottom curve in each set of 4 is on the ground. That one, that one, that one, that one. And then over time in space. So let's just start with this one. This was in contact with silica and argon ambient. This is isobioanthene, which is this molecular chicken wire, as we chemists call it. And this is September 2010. This is November 2010. Hasn't changed much. This is September 2011. So this is after a year in space, and it has not changed much at all. Maybe a little degradation. Pretty much true for this one, which was also argon, but without the silica contact. Pretty much true, though there's a little degradation here with some CO2 and a little bit of oxygen. This one with water, it does take a while, but it's pretty much nearly gone after a year. So why is that? Well, water makes hydroxyl radicals when you shine 10 electron volt photons on it, and hydroxyl radicals eat organics very effectively. So the UV alone was not enough to destroy this organic molecule, but UV plus some humidity was. And so this is kind of interesting from a perspective of how long might organic molecules hang around on an asteroid if they got, at least part of the time, some exposure to sunlight or starlight. And the answer is a lot less time if there's any source of water around, as you find in the comments. If it's a really dry environment, they might tolerate lots of UV before they degrade very much. Similar story for iron tetraphenyl porphyrin. Somewhat surprisingly, I would have thought it would be quite a lot more sensitive. But in the pure argon with a bit of oxygen and some CO2, or pure argon with nothing else, you get about the same response. This is over the same time course, about a year. Actually, no, this is up to 17 months. And there were some subtle changes in here, which actually may have to do with this very low concentration of oxygen, I'm not sure, or some trace water. But not a lot of degradation. And then with the relative humidity, it degraded significantly. No, it's not necessarily all gone after 18 months. Okay, so that's the end of the story of OREOS. The conclusions are that you can do some pretty interesting science in space with small satellites. We're all developing the tools, the sensors, the microfluidics, and the rest of the technologies that make this happen, the batteries, all of it. You need to be good at integration and automation, and you need a really good systems engineer to do this. One of the things we do that they don't do very often on the ISS is we get real-time in-situ measurements. So we can follow the kinetics of molecules changing. We can follow the kinetics of biology growing. A lot of these other space experiments, they take them up in space, they leave things for a while, they freeze them, they bring them back and analyze them. So they get T0 and endpoint. We get all the time in between. So that's more interesting. And that's a motivation. People say, the space station's there's lots of flights, why don't you just go to the space station? Well, one reason is, although there are lots of flights, it's often very well booked. The other reason is we can get kinetics off this that they often can't get on ISS. And there are a lot more launch opportunities, and this can cost less. I've heard people say it costs $1 million to send a brick to the ISS. I don't know if that's really true, but we spend our $1 million more wisely than sending a brick. So this was all been funded by NASA. The OREOS mission I told you about for most of today's talk was funded by the Science Mission Directorate. Both GeneSat and PharmaSat that I mentioned at the start were funded by the Human Exploration and Operations Mission Directorate. Actually, it used to be 2 other directorates, but that gives you a rough idea. So thanks to them for the funds, and thanks to all of you for your kind attention.